Hi everyone, we're group 7 and we're going to be presenting about the engineering pathogen resistance in crops. So a quick introduction to plant pathogen interactions. The plant cell wall serves as the first level of defense against pathogens. Bacteria can be sucked into the plant through natural plant openings such as the stomata, hydrothodes, or lenticels, or through abrasions on wounds on leaves, stems, and roots. Plant pathogen interaction was first discovered as a gene-to-gene model. This model explained that with every resistant gene in the plant, there was a matching one in the parasite. It was found that the resistant genes work in coordination with each other to build the plant's defense system. This was evidence that plants have a genetic defense system and a physical defense system against pathogens. With further research and advancement of technology, it was discovered that plants can build resistance with genetic engineering, traditional breeding, marker-assisted breeding, and mutation breeding.
So there are multiple examples of genetically engineered pathogen resistance that are present in the world today, and I'm going to talk about two of them right now. There is an example of late blight fungi, which is caused by Phytophthora infestans, which is a potato blight. And there's a resistant gene RPI VNT 1.1, which was isolated from the wild potato species Solanum venturi to provide resistance for this disease. There's another example that is banana xanthomas wilt, also known as BXW, and it is a bacterial disease caused by Xanthonomus campestris, PV musacarium, or XCM, and resistant strains of hypersensitive response assisting proteins, or HRAP, and plant ferredoxin-like proteins, or PFLP, from sweet pepper have been extracted and were expressed in bananas to provide resistance to BXW. Below are photos of the potato blight and the bananas infected with the Xanthomas wilt.
Another significant example of a pathogen-resistant genetically modified crop is papaya, which was genetically engineered to be resistant to papaya ring spot virus, or PRSV. PRSV is non-persistently vectored by aphids, and it poses a massive threat to papaya growers in the US and worldwide, especially causing significant loss of up to almost 50% from 1992 to 1999 in Oahu, in Hawaii, where growers attempted various methods of defense, including eradication and other cultural controls to chemical spraying, but was ultimately unsuccessful.
In 1986, scientists found a way to make the papayas immune to PRSV, and they did this by looking back on what had been done earlier in tobacco crops. So by expressing the biocoat protein to the virus affecting tobacco in the crops, they were able to confer immunity to the tobacco. So the scientists looking at PRSV in papayas did the same, and by expressing the biocoat protein to PRSV in papayas, they conferred immunity to the disease. And then these papayas wound up becoming the first commercialized transgenic crop that had innate pathogen resistance.
So we are also going to be taking a look at 3 non-genetic engineering methods. Those 3 are traditional breeding, which is breeding the crops for desired traits, marker-assisted breeding, which is placing tags on genes, and mutation breeding, which is exposing plants and seeds to mutagens in order to create mutations.
So beginning with marker-assisted breeding, this is a variation on traditional breeding that uses DNA markers associated with specific traits, and it's used to select individuals during a breeding experiment. So this is a really important technology because it can greatly reduce the amount of time required to identify individuals that express ideal traits. And it tends to be a little bit more specific as it relies on genotype instead of phenotype. So the figure on the right is an illustration of this concept using wheat. So the DNA on the left represents a parent with a marker for dough strength, and the DNA on the right represents a wheat parent with marker for stripe rust resistance. These 2 are bred together, and then the progeny can be screened, and if one of the progeny has all 3 markers, or all 4 in this case, then it might be an ideal crop. An example of when this has been used and commercialized is in apple scab. So apple scab disease is caused by Venturi iniquialis and is persistent anywhere that apples are grown. It thrives in a cool, moist environment, such as that as the environment in early spring, especially in New England. So it causes lesions on fruit and leaves that can cause large crop losses. This disease drives one of the greatest fungicide use in the Northeast in apples. So it's a really big problem for farmers and a really expensive problem as well. The images on the right show pictures of lesions on leaves, and then on to the right of that is a picture of lesions on the apples. So the experiment that was done using marker-assisted breeding was involved 3 different genes. Initially, it was thought that apple scab was caused by just one gene, and traditional breeding methods have been used since 1945 to confer resistance to apple scab. But Baumgartner and his colleagues targeted RBI6, 2, and 4, creating breeding lines that were homozygous for the resistance genes. And the table on the right shows the results of this experiment, and it showed that all 3 genes, when they were combined, had greater resistance to apple scab. So the columns represent the infection class from 0 to 4, with 0 being the least degree of infection and 4 being very severe infection. And so all 3 resistance genes combined had much lower rates of significant severe infection.
The last type of breeding is the gold pair. This type of breeding is mutation breeding, which has radiation, which changes the DNA, and it can either cause an early stop codon to appear, or can insert a codon which causes a new protein to form. The particular pair that we looked at was the Japanese pear, which was very susceptible to black spot disease. Many young pear trees were treated with chronic gamma radiation, and the mutated twigs were identified at the 40-meter mark, and these twigs were grafted onto mature Japanese pear trees. As you can see on the right-hand side, the diagram shows the experimental setup going from 40 to 70 meters with radiation exposure. GMOs, yay or nay? Our group particularly thinks GMOs is a good thing. A study published in 2014 showed that crop increased yield by 22%, increased farmer profits by 68%, and the last 20 years reduced chemical pesticides by 37%. U.S. customers have experienced no cost or nutritional benefit from GMOs, and many people wonder what their risk and environmental impacts are. But for many developing countries, GMOs is a good alternative to being able to produce food that is viable for many countries who do not have either good farming techniques or the land to farm. Like for an example, like rice over in China, that when their fields get flooded, people are trying to find ways to genetically modify rice so that it can be flood resistant. We'll take any questions at this time, and thank you for watching.